Up to this particular point, we've talked about the amount of neurotransmitter that's released from a presynaptic neuron as being constant in every single situation. And it's perhaps necessary to do that to begin this discussion, but we want to point out now that that's absolutely not the case. But interestingly, a single presynaptic neuron can vary tremendously in the influence it has because the amount of neurotransmitter that can be released from a presynaptic neuron can vary widely from one stimulatory event to another. Now, I do want to clarify, that doesn't mean that we are changing the nature of the influence of the presynaptic neuron. As we said previously, a presynaptic neuron is either excitatory or inhibitory, but it's not both. All we're saying in this case is that the amount of either excitation or inhibition that can be applied by the presynaptic neuron varies from stimulatory event to stimulatory event. So why is that? Well, as we see on the slide here, the number one reason would be that the amount of calcium that's available in the presynaptic cell can vary widely as we look at one stimulation event versus another one. As we previously discussed, it's calcium that binds to synaptotagmin and then interacts with those SNARE proteins, causing the fusion of that vesicle containing the neurotransmitter with the presynaptic membrane. So this means that if you alter the level of calcium present in the presynaptic cell, you're going to alter the number of events that I just described there, and therefore the number of vesicles filled with neurotransmitter that are fusing with the presynaptic membrane. Now, why would calcium levels vary from event to event? Well, the main reason would be this, that we simply have calcium that builds up between successive stimulatory events. In other words, between successive action potentials that reach the axon terminal. So one action potential arrives, calcium is released, and then the whole event with the fusion of the presynaptic membrane with the vesicle occurs, and then calcium levels start to go down. But if another action potential arrives before all the calcium has been pumped back out of the cell or into intracellular organelles, then you're going to have this summation of the level of calcium found in the presynaptic terminal there, and therefore you're going to have a greater concentration of calcium and more neurotransmitter released, and therefore, obviously, you're going to have a greater influence on the postsynaptic cell. So the number one reason that calcium levels would vary is a buildup between successive signals, in other words, successive action potentials. Another possibility is this, that you have an axo-axonic synapse, and that's what we see depicted in this particular slide. So we have three axons depicted here, three neurons depicted here. We have A, the axon terminal of neuron A, the axon terminal of neuron B, and then we have the cell body of neuron C. And so A is influencing B, B is influencing C. This, the junction between A and B, between these two axon terminals, is referred to as an axo-axonic synapse. And this can lead to presynaptic inhibition or presynaptic facilitation, primarily via one of two mechanisms. One possibility is that a neurotransmitter is released here from A, diffuses across this cleft, if you're following the pointer, it binds to the receptors that are found on B. These are ionotropic receptors, and so they open, allowing calcium to rush into the cell. And so not only do we have an increase in calcium levels as a result of the arrival of the action potential down the axon of B, but we also have calcium that's entering as a result of the influence of axon A on B. Another possibility is that A releases a neurotransmitter, just like we described, but the receptors to which they bind are metabotrophic, and so they utilize second messengers to bring about an increase in neurotransmitter synthesis. So there's not an altered level of calcium, but what you do have is simply a higher concentration of neurotransmitter. So when the calcium arrives as a result of the active potential coming down B, more calcium is going to be released. A final possibility for the variation in synaptic strength is something referred to as autoreceptors. And so autoreceptors are shown here, 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 and here. And autoreceptors, interestingly, bind to the neurotransmitter that's released by the presynaptic cell, influencing the calcium levels or the neurotransmitter synthesis within the presynaptic cell. This would be expected to be a more minor impact, but could influence the amount of neurotransmitter that's released from this particular neuron. And so, for instance, if this neuron doesn't work for a while, if no action potentials come down it, then no neurotransmitters are released and no neurotransmitter binds to these autoreceptors. But if action potentials frequently make their way down B, then that would mean that these autoreceptors are constantly finding a ligand, and so they are providing stimulation to this axon B and making it easier for subsequent action potentials to release neurotransmitter at this cleft, resulting in a greater effect on the postsynaptic cell here. So the overall point is this, from this particular slide, that the amount of neurotransmitter released by a single presynaptic cell, which would be B in this particular case, is not constant from one event to the next. Not only can the amount of neurotransmitter released by the presynaptic cell impact the strength of the signal that is carried across the cleft there, but interestingly, the strength of the signal can also be modified by the postsynaptic cell. So in other words, at any given level of neurotransmitter released, the postsynaptic cell can respond very strongly or fairly weakly to that stimulus that is released. The most common reason for this would be different receptor types. There are many different receptor types, as we have mentioned, for neurotransmitters that are found on the neurons in primarily the central nervous system, but also to some extent the peripheral nervous system. And there are even many subtypes of these receptors that have a very profound influence on the response of the postsynaptic cell. So some of these receptor types have a greater influence than others, and some of these may even have opposite effects. The most obvious example, and one that we'll talk about very soon, the most common example most likely, would be receptors for acetylcholine. And so we'll see that nicotinic receptors always excite, but muscarinic receptors can inhibit. They can also excite, but they frequently lead to inhibition of the postsynaptic cell. So same neurotransmitter, but opposite effects on the postsynaptic cell. Another possibility, and this is similar to what we see in other areas of physiology, is that the cell can regulate the number of receptors that are present for that particular neurotransmitter. And so up and down regulation of receptors is very common and would have a very profound impact on the response of the postsynaptic cell to any given concentration of neurotransmitter. Lastly, a receptor that is continually stimulated by a particular neurotransmitter can interestingly eventually not respond to that particular neurotransmitter. This is not always possible, but certain receptor types undergo this phenomenon referred to as receptor desensitization. This is something that's sometimes observed in the abuse of illegal or prescription drugs when the individual that is using these drugs has to use an increased amount of this substance to achieve the same result over time as a result of the fact that the receptors that on the neurons that respond to this particular drug are becoming desensitized. Here we talk briefly about the impact of drugs and disease on synapses. And here we see this fascinating discussion where the complexity of this system that we're learning about provides multiple opportunities for both synthetic and so-called natural, in other words, produced by organisms in nature, compounds, to influence this system. And there is some overlap between drugs and disease, as we'll see just in a simple example in the next few moments. So obviously you're aware of the fact that botulism is a very serious disease caused by food poisoning, but in modified amounts, that same toxin is used, of course, for what we know as Botox. So we will build on this discussion moving forward. We don't have a lot to say at this particular point regarding drugs, but we do want to take a look at this particular slide and make some notes about the potential places where either drugs or natural compounds that cause disease could influence this system. And so we have a brief list on our screen. These are mostly things that you are familiar with at this particular point. But let's go through them briefly. So what we see is this, that a drug or other compound could cause increased leakage of neurotransmitter from vesicles, and therefore it could be destroyed by enzymes, and you'd have lower levels of that neurotransmitter in the presynaptic cell. B, you could increase, as we see here, the drug could increase the release of the neurotransmitter into the cleft. C, obviously, the drug could block the release of the neurotransmitter into the cleft. D, the drug could inhibit the synthesis of the neurotransmitter. And let's see, E, and this is fairly common, the drug could block the transmitter reuptake, and so SSRIs would be a good example of such a drug, serotonin-specific reuptake inhibitors. F, what we see is that we could block the enzyme that's found here that breaks down the neurotransmitter. In other words, the drug could do that. That's also relatively common, and so disturbingly, something like the nerve gas that we talked about previously in class that was used in the assassination of the Korean leader's half-brother, that's exactly what was used. Those nerve agents are very dangerous and fast-acting compounds that do just that. They inhibit acetylcholinesterase in this cleft. G, we could have a drug that binds to, or a toxin that binds to, the receptors that are found on the postsynaptic side of the cleft. We do want to pause for just a second here and make sure we remember that at this point, two things could happen. One possibility, and this is outlined over here and in the bullet points there. In other words, two things could happen. One is that the neurotransmitter could bind and block these receptors. It binds and does not have an effect. That would be an antagonist. Or it could bind and have an effect, just like the natural ligand does. In that case, the drug or toxin would be considered an agonist, as you know. And then lastly, we could have a drug or toxin that inhibits or stimulates second messenger activity within the postsynaptic cell, which may perhaps be a little less common, but also observed in some cases. So let's look at a couple examples of drugs that impact the synapse. Interestingly, certain types of bacteria, especially the genus Clostridium, produce a number of toxins that have a very profound impact on this synapse. So one of these, and the first listed on this slide, is the bacterium Clostridium tetani, which produces a toxin known as, your book labels it as tetanus toxin, sometimes referred to as tetanospasm. And this particular toxin interacts with the system that we just described by destroying the SNARE proteins and preventing, therefore, the fusion of the vesicle with the presynaptic membrane. Interestingly, the effects of this toxin are specific and they only impact inhibitory neurons. So the end result is that we have two impulses going to our pairs of muscles at any given time. In other words, let's use the arm as an example, the upper arm. So a stimulatory signal would be going to both the bicep and the tricep simultaneously. And when both of those contract at the same time, you end up with this so-called spastic paralysis. And so an extreme example of that is illustrated in the painting that we see to the right, a very famous painting, unfortunately, of an individual in the later stages of tetanus. This spastic paralysis and these very violent contractions get worse and worse and worse until eventually vertebrae break during these contractions, and death comes as a result of the inability of the diaphragm to contract and relax. Fortunately, in developed countries, vaccine is widely available for this particular toxin. It's very disturbing that in developing countries, although this vaccine is not expensive or difficult to distribute, it's largely unavailable in many, unfortunately, parts of the world today, and tetanus is a big problem. Now, we go down to botulism, and so this is another little bad bacteria from, at least for humans, from the genus Clostridium, Clostridium botulinum. Clostridium botulinum produces a toxin known as botulinum toxin that's one of the most, if not the most potent toxin that's known to man. And so apparently the volume or the weight of toxin that's in the lead in a period that you would make with a pencil on a piece of paper is enough to kill a huge number of people. It's extremely potent. And this particular toxin also destroys SNARE proteins, but unlike the tetanus toxin, it targets excitatory synapses. And so the end result is death as a result of simply paralysis, not spastic paralysis from the contraction of muscles, but rather just paralysis from the failure of muscles to contract. And like tetanus, death eventually comes as a result of the inability of the diaphragm to contract and therefore the animal, person or otherwise, to breathe. So both of these are interesting, although somewhat disturbing, admittedly, examples of toxins produced in the natural world that have a profound impact on this synapse, the system that we're learning about at this particular time. These are also, I should point out, very common bacteria. You could go out on campus today, get a soil sample and locate most likely, certainly tetanus and likely Clostridium botulinum in that soil sample today. Interestingly, what we see is that, not only is it bacteria, but there is a long list of organisms in the natural world that have evolved molecules that interact with our nervous system. And it's not our role here to go through an exhaustive list, but it is something that's interesting to consider. So on the screen, we have a black mamba. Black mambas are one of the most venomous snakes in the world, and no one was, in recorded history, survived a black mamba bite up until the late 1960s when antivenom was available. And they produce a toxin known as calciseptine. And so this is a neurotoxin that interestingly blocks calcium channels. And so you're, of course, familiar with the role that calcium channels play in the transmission of a signal from the presynaptic to postsynaptic neurons. So, of course, they cause paralysis as a result of the inability of calcium to enter that presynaptic neuron, and therefore, the failure of calcium to interact with synaptotagmin and SNARE proteins, and so the neurotransmitters never release. So this also brings us to an interesting point, and that is that these toxins that are produced in the natural world by these organisms or bacteria oftentimes have very valuable applications within the world of medicine. And so the person we see on the screen there is Jim Harrison. He owns the Kentucky Reptile Zoo, which is located in Slade, Kentucky, near the Red River Gorge. And they have one of the biggest collections of venomous snakes in the world. He's holding a cobra there, and they extract venom from these snakes and sell it. And there's this tremendous demand for this venom by researchers who rarely would be interested in extracting the venom themselves and maintaining these snakes. Because of the interesting ways that this venom, or toxins for that matter, if you expand the discussion to amphibians, interact with the nervous system of humans.